will only evaluate part 2, okay, so the material that ends today. So that means the next lecture on Friday will be on part 3. We'll start part 3. That material forward will not be on midterm 2. Okay, midterm 2 doesn't happen until, I believe it's the 8th. It's a week from Wednesday of next week. So we have ample time. What's that? The 6th? Okay, the 6th. And so there will be ample time for us to review that material. We're ahead of schedule, and that means I'll do a review session, but not until Monday, the lecture prior to the midterm, so that you have as much time as possible to study. So that gives us a whole office hour next week. You guys can come with questions on part 2 lecture material. Ample time to study for the test. Okay, so we left off with the digestive system, and what we've covered thus far is some of the mechanical digestion, and just for context, we're following a cheeseburger, the life of a cheeseburger through your GI tract. And we've done swallowing. We covered some basics on peristaltic waves and how you have these layers of muscle that drive peristalsis, but also churning. And so now that we've covered those basics, let's consider the travels of this cheeseburger. And so those travels begin with the esophagus, which we've already mentioned. And then, of course, the esophagus dumps its contents into the stomach. And so we have the stomach right there. Right there, there's the stomach. And of course, the stomach is isolated from, it has very low pH, and it's isolated from the rest of the GI tract by muscular valves called sphincters. And so we have sphincters up at the top of the esophagus that keeps water or other liquids from backflowing as you begin to swallow. And also, they seal off that high pH, low pH that's within the stomach. And when this sphincter fails to do its job, then you get acid reflux, and eventually you can burn a hole in the wall of the esophagus. That's an ulcer. Okay, so the cheeseburger makes its way into the stomach, and the stomach has not just 2 muscular layers, but 3 muscular layers, and it goes through all kinds of contortions, and we're used to feeling that kind of churning and groaning of the stomach, and that's partially the muscular activity as it's processing your cheeseburger. And so there's a variety of motions that these 3 muscle layers are capable of, given a very localized contraction, very localized activation of those muscle layers. So there's this forward and backward churning of the material. So you can imagine if you have a little pinching, like as pointed out by those arrows in one spot, then that's going to propel food away from that little spot. And then there's also, versus pinching at a slightly different spot will, of course, propel it in the other. So you can have the food sort of sloshing back and forth as a consequence of pinching different regions of the stomach. It's also capable of sort of grinding up the food. So this is what happens when you haven't chewed your meal sufficiently, then your stomach has to do the rest of the work to break it down. So that's one feature of the stomach. It's another muscular organ, like the heart is a muscular organ. The stomach has muscular walls, and they're doing this all the time. At the same time, so it's basically finishing the job of the mechanical digestion that you failed to do before you swallowed the cheeseburger. The rest of it has to do with chemical digestion and also the killing of bacteria that comes with having this low pH environment. And so let's zoom in on the walls of, or the wall of the stomach to consider some of the cell types that are responsible for doing this chemical digestion. So we have a layer of cells, the epithelial cells on the inside, and just bear in mind, like with a lot of epithelial cells that we talk about, they're well vascularized, and so you have a capillary and close proximity. And the first cell type that I'll mention is the one that's responsible for the low pH. That's known as a parietal cell. And then another important cell type is known as a chief cell. And chief cell secretes an enzyme known as pepsinogen. Okay, so we'll get into what that does in a minute. The parietal cells, by secreting hydrochloric acid, create the low pH. Okay, so there are two big functions that I'm going to emphasize on that. Is first of all the killing of bacteria. So we have some enzymes in your mouth that will begin the process of killing bacteria, but to really get rid of the bacteria, most of them will die in the stomach due to this low pH. At the same time, that environment activates proteases. And the pepsinogen that is secreted by the chief cells in particular are activated by this low pH. Now, the epithelial lining is composed largely of proteins. And so if you have a protease that has a job of breaking down proteins and a low pH environment, then you might ask the question, well, why is it that the stomach wall doesn't just digest itself? How is it that they're kind of magically protected from the things that they secrete? So there are a couple of parts to this story. The biggest component or the biggest answer to this question is mucus. So we have what are called mucus neck cells that secrete a particular brand of mucus on the interior of your stomach. And as a consequence of what they do, we have a mucus layer that serves almost like a force field for those epithelial cells to protect them from low pH. And this is no ordinary mucus. This mucus is very rigid. And so you can think of this mucus as almost being like a like snow. And you can imagine in a blizzard with a large snow accumulation that you've got a structure of snow in a way. And so the mucus does that. It's essentially burying these villi, the finger-like projections of the stomach lining, in a snowy mucus barrier. And as a consequence of this, the pH that's inside of this mucus barrier is something that's like a healthy pH for most cells, 7.2 for example. Whereas up above, we're showing the lumen up here of the stomach. This is the kind of environment that is going on in the center of the stomach, a very low pH. Okay, pH scale is, of course, logarithmic, and so we're talking about orders of magnitude more acidic in the center of the stomach than in the mucus layer. All right, so then you've got a problem like, all right, we've got this force field, but somehow we want to dump these toxic chemicals into the interior of the stomach, and they have to traverse this mucus, snow-like accumulation that they're buried in. And so the mucus buffers against the hydrochloric acid. Okay, also another thing is that pepsinogen is by itself not toxic. Pepsinogen is in a non-toxic form, and it is pH activated. So pepsinogen will be activated when it can reach its way into the center of the stomach. But as long as it's down in the mucus layer, then it's not toxic, which is fine for the epithelial cells, but it also isn't active. It can't break down any large proteins either. So at high pH, pepsinogen is inactive. Okay, so how is it that we get these toxins transported into the lumen of the stomach? Okay, the answer to that is in the form of what are called mucus fingers. So imagine you're buried under a layer of snow and you want to get out. You're going to burrow in through the snow and make a tunnel that allows you to escape. And that's essentially what happens, is the high pressure generated by the parietal cells and to a lesser extent the chief cells create these fingers through the mucus that creates essentially vessels or tunnels. And these tunnels allow for the transport of the hydrochloric acid. So it's not like a capillary that has walls around it or anything like that. It's like an absence of mucus. It's a little vessel is the word I was looking for. It's a little vessel that goes through this layer of mucus to allow for the hydrochloric acid to find its way to the lumen of the stomach without dissolving the cells along the way. And since the pepsinogen is transported in a similar way, then it also doesn't begin to react until it makes its way into the lumen. And so it's in the lumen that the pepsinogen becomes toxic. And it becomes toxic by converting into pepsin. And pepsin breaks down internal peptide bonds. Okay, so that's sort of the Achilles heel of a large protein. If you start breaking down those peptide bonds, then you go from large proteins into much smaller proteins that are more digestible, which is therefore going to help on the digestive process, as well as potentially any proteins for any pathogens that made their way in as well. Okay, so the way that the stomach doesn't digest itself is through the mucus layer, and it's still capable of transporting these toxic chemicals via those mucus fingers. So that brings us to a clicker question. What symptom would you expect in the scenario that you have an autoimmune disease that degrades the chief cells? So the possible answers are A, a low concentration of glucose in the blood, B, a high protein content in the stool. Okay, that means that gets pooped out of your body, it's defecated. C, a low concentration of triglycerides, that's fats, in the blood. Or D is all of the above. Okay, give me your final answer on this question. Okay, so the correct answer is B. So 80% of you got the right answer on this question, but this question is pretty typical of digestive system multiple choice questions. So what it requires is you need to know what a chief cell is. Then you need to realize that, oh, if I don't digest something so that it can be absorbed, then it's never going to enter into the bloodstream. It will end up in your stool, or the question might ask, or a possible answer might be defecated or something like that. So anything that does get absorbed is potentially going to be reflected in your blood concentration. So it's just good to keep that in mind for most of these digestive system questions. So if it's not going to get broken down, or if it's not going to get absorbed, it's going to get pooped out. And anything that aids in either the digestive or the absorptive process could be reflected in the bloodstream. Question?
So if we know that proteases break down proteins, what breaks down like glucose?
We'll talk about that in a second.
Oh.
Yeah, don't worry. We'll cover it all. Yeah.
Just to clarify, with the peptides from the pepsinogen, how does it break into peptide? How does it break into peptide?
Peptide bonds?
Yeah.
So here's that slide again. So how does it do it? Well, through the magic of enzymatic action. So this is an example of a situation where we largely skim over the details and just give a black box explanation. But it has to do with the biochemistry of enzymes. So just imagine that there's a large molecule, you have peptide bonds that are keeping it together, and the enzyme has a means of cleaving those bonds. And that's the only level of detail that I can provide and that you need to know.
So basically, the enzymes in the stomach break it apart?
Enzymes in the stomach begin this process of protein digestion, chemical digestion.
Okay, thank you.
Yeah. So we've got the mechanical digestion going on in the stomach. That's taking still big chunks of cheeseburger. And then at the same time, the proteins, and cheeseburgers have lots of proteins, the proteins are getting broken down at a molecular level through the cleaving of the peptide bonds. Okay, so the broad context looks something like this, that we have this chemical digestion. So now in the stomach, we're sort of handing off between mechanical and chemical digestion. Essentially, by the time the cheeseburger emerges from the stomach, all the mechanical digestion is over, and the chemical digestion can proceed. So by that point, you still have large molecules that many of which might not be capable of getting absorbed. And so it's a process of chemical digestion to make those smaller molecules to allow for absorption. Okay, so that brings us to these organs. The liver. So the liver kind of gets neglected in this course because we don't have any dedicated liver lectures. But we'll see that it plays a role in the digestion of fats in this case, and then we're going to see later on how it plays a big role in energetics in general. So the liver's an important organ. The stomach is connected to the small intestines. So the small intestines are actually a lot longer than the large intestines, but they're small in diameter. And then eventually the meal will make its way into the large intestines, where we'll have a lot of water absorption. That's where bacteria are housed. They'll do the final processes of chemical digestion, and the storage of the feces occur in there, in particular in the rectum. So by the time the meal makes its way there, then you feel it and want to get rid of it. And in between all of these passageways are more sphincters, more smooth muscle closures. And in the case of the rectum, you actually have skeletal muscles. So sphincters can come both in the variety of smooth muscles and skeletal muscles. And we like skeletal muscles because we can control them. And so fortunately, we can control defecation. All right, we'll get to that in a minute. For the small intestines, okay, so this is just what happens right outside of the stomach. So we have a sphincter that, and by the way, if you ever want to take gross anatomy, you can learn all the names of the sphincters. They all have their own special names. And the different regions of the small intestines also have special names, rather funny special names. The first section is either pronounced the duodenum, which is how I pronounce it, or the duodenum, which is the way some people pronounce it. And then there are a variety of sections after that. We won't put labels on those. And then eventually there's another sphincter that goes between the small and large intestine. And so I'm going to lay out, in the duodenum, you do have these secretions that only occur right there because of the pancreas and the gallbladder, which is where the duodenum's situated. But then after that, you have absorption. And absorption occurs, and digestion for that matter. All of those processes just continue through the entire length of the small intestine. So I'm going to explain these details of what occurs at the duodenum, but just imagine that that just keeps on happening as the meal gets propagated through the small intestines. And as a consequence of there being more food at the beginning, you have high absorption right there at the duodenum, and progressively less as there are diminishing returns on the meal. But fortunately, the small intestine is very, very long, and so we squeeze a lot of nutrients out of that potential meal, or out of the meal. Okay, so let's zoom in on this juncture between the stomach and the duodenum. And there's a couple of problems that need to be resolved by the duodenum. And first, a little bit of cellular anatomy. Let's focus in on the wall of the duodenum and the small intestine in general. And this wall is called the gastric mucosa. And it has not just an epithelial barrier, but there's a bunch of connective tissue. It's surrounded by the smooth muscles that generate peristalsis to propagate the meal. And if we zoom in at a cross section, it looks something like this. That's the epithelial lining. You have little finger-like projections. Those are called villi. And then little cavities. Those are called crypts. And throughout, it's well vascularized. So you can just assume that there are capillaries. And in addition, there are these little vessels that are analogous to capillaries, but they're part of the lymphatic system. And those are called lacteals. Okay, so lacteals, as we'll see later, transport fats. That's their role in digestion. So instead of fats like fatty acids and glycerol making their way directly into the capillaries, they get transported through the lacteals. All right, in terms of cell types, along the gastric mucosa, there are a few. We laid out some for the stomach. We have some others for the rest of the GI tract. We have absorptive cells, and so when we talk about the absorption that goes on for proteins, fats, or carbohydrates, they're going to occur across these cells. And they look like this little Bart Simpson head with little further finger-like projections. And so wherever you have a little finger, you can assume it's got a lot larger surface area than a flat surface. And so this is a villus, a finger, on the gastric mucosa. And then each of the cells has an apical surface with further little fingers. Those are called microvilli. And this arrangement of microvilli on the apical surface is sometimes referred to as the brush border. I apologize for all the jargon. In the digestive system, I have subtracted the jargon each time I teach the course, but I still can't dispense with these terms. So I'll mention what the brush border does in a moment, but they are anchors for enzymes. So there are enzymes that are just sort of dumped into the lumen of the small intestine, just like the pepsinogen that's dumped into the lumen of the stomach. But alternatively, on the brush border, you have enzymes that are holding on to the brush border and cleaving molecules on domains that are capable of doing that. So they never get sort of swept out through the system. They're anchored to the system. Was there a question?
Yeah, just want to clarify. So brush borders are a bunch of microvilli, or?
Yeah, brush borders are an arrangement of microvilli. And the brush border in general, it's also referring to the fact that it's the apical side of that cell. It's the side that sticks into the lumen of the gut.
Okay, thank you.
Okay, so we got the microvilli, or I'm sorry, we've got the absorptive cell. We have cells in the small intestine that also secrete mucus. Those are called goblet cells. And then we're only going to touch on a little bit of it, but there are a variety of hormones that are secreted that only matter to the internal functioning of the stomach. There's a lot of decision making that goes on in the stomach. There's a lot of sensing. There's a lot of responding. Because as you can imagine, since your diet can vary so much in its composition, and the particular enzymes that are going to act on that meal are going to vary depending on the meal, not to mention the size of the meal, a mixture of its components, how acidic it is, all of those variables. If you want to get as much nutrition extracted as possible, then you want to have a very intelligent digestive system. And in fact, there's a whole network of neurons that makes decisions called the enteric nervous system, which we're not going to really talk about in detail. And it's also a little bit mysterious about how it works, but it's almost like a brain for your gut. And so there's a lot of decision making that's completely outside of the central nervous system that just goes on in the gut. And part of the responses to the chemistry of a meal are mediated by the endocrine cells. They will secrete hormones that then have an effect on the motility and the chemistry of the meal as it gets propagated through the system. All right, so absorptive cells, goblet cells, and endocrine cells. All right, so by the time the food emerges, so the burger emerges from the stomach, it's really a mess. You can't even identify it anymore. It's an acidic food slurry. So it's acidic because it just emerged from the stomach, and it's a slurry because all the chemical digestion or mechanical digestion has been completed, okay? So that's going to be a lot of large molecules in a very sort of gross mass. And so there are a couple problems that need to be resolved here. First of all, it's acidic. And then secondly, there has to be the ability to recognize the nature of this meal so that we can control its absorption. So let's consider a couple of solutions to these problems. So first of all, neutralizing the stomach acid. Now, first of all, those endocrine cells are sensitive, or they're pH sensitive. And so a certain class of them are going to respond to the low pH of this chyme. Chyme is such a vulgar word, but it seems appropriate. So the chyme activates the release of a hormone called secretin. And so whenever I say that a hormone is secreted here, just imagine that it's not just in the lumen of the gut affecting the epithelial cells on the inside of the gut, but also being transported through the bloodstream and affecting any organ that has a receptor for secretin. And so those receptors include cells that regulate the secretion of acid in the stomach. Okay, so the reason for that is you've got this acidic food emerging from the stomach, and the low pH environment is only ramps up when there's a meal in the stomach. It's not something that's maintained, at least if you're healthy, maintained all the time. It's energetically expensive to maintain that, to be secreting the mucus and everything. And so as soon as the food starts emerging from the stomach, then the secretin is effectively sending the signal to turn it down, okay, to reduce the secretion of the hydrochloric acid. Okay, another big organ here is the pancreas. And so the pancreas is a gland that secretes bicarbonate and digestive enzymes. It also plays a big role in regulating energy balance, which we'll talk about later. And so secretin also acts on the pancreas. And in particular, it's the pancreas that's responsible for neutralizing that low pH of the chyme. And it does that by secreting bicarbonate. And it releases that bicarbonate into the small intestine. And so by the time the chyme makes its way down to this region, we have this ability to neutralize the low pH. So the low pH is, of course, a reflection of all those protons in the solution. And provided there are chloride ions around, then the bicarbonate then creates carbonic acid and table salt via this reaction. So just dumping bicarbonate into the lumen of the stomach is the pancreas's solution to lowering or raising up the pH. And that's good because low pH would burn a hole eventually in the small intestine, and so this is neutralizing that from happening. Okay, at the same time, we have the pepsin. Remember, the pepsin is breaking down peptide bonds, and so that presents another toxin. And so by raising up the pH, not only are we making it so it's less acidic for the rest of the GI tract, but we're also going to be neutralizing the pepsin. Okay, yeah, question?
So secretin, does it neutralize the acidic part by itself, or it needs to use the bicarbonate from the pancreas to do so?
The bicarbonate is what neutralizes that low pH. but it's being triggered by secretin. So secretin's role is to essentially alert both the stomach and the pancreas to this low pH situation so that less acid is secreted by the stomach and bicarbonate is released.
Thank you.
Okay, so the other hormone I'll tell you about is also released, and it is sensitive to fat. And so fat takes longer to absorb, and the goal here is to slow the peristaltic waves that propel the chyme through the small intestine. And so the hormone that is used for that, I always pronounce this a little bit wrong, cholecystokinin, or we'll just call it CCK. So CCK gets released if you have eaten a fatty meal. And the main response is that, well, the first main response is that it reduces the motility. So all that smooth muscle that has its spontaneous contractions and its decision making on its own, all that is going to slow down if it gets the CCK signal. Okay? And so that's an example of a response where the stomach is being intelligent with regards to the meal that has been eaten and changed its behavior accordingly. Question?
So people want to go on keto diets, have release a lot of CCKs.
I'm sorry, what kind of diet?
Keto diet, ketogenic diets,
I don't know what that is.
high fat diet.
High fat diet, right.
So
Right.
they're most likely to release a lot of CCKs.
That's right. Yeah. Yeah, CCK is the stomach's, well, it's an example of an endocrine response to a diet, or a variable that matters in the diet. And it slows down peristalsis. Yeah. Okay, another thing that it does. So I mentioned that the liver plays a role in fat absorption. And so the way that it does that is through bile salts. And so the liver secretes bile salts, and they're held within a bladder, a muscularized sac. And that bladder, like everything else here, has a smooth muscle lining. And those smooth muscles respond to a CCK signal. So as CCK rises in concentration in the blood, it causes tension in the walls of the gallbladder, which elevates the pressure, and that pushes the bile salts or slurry of bile salts out from there, and then it follows this channel down here into what's called the common bile duct. And so the bile also, along with bicarbonate, is emerging from this common channel that's also connected to the pancreas. And so that bile is coming from the gallbladder, which originates from the liver, and then gets dumped in the center. So that's going to play a role when we talk about the absorption of fats. Just keep in mind that if the meal is fatty, then bile is released by the gallbladder in response to CCK. Okay, at the same time, the pancreas plays a role in releasing enzymes and water, again, a response to the release of, to the fatty content of the meal. We'll see there are enzymes that play a role, in particular, in breaking down large fat molecules into smaller ones that can be absorbed. Yeah, question?
Is this specified for unsaturated fats versus saturated fats?
This would be the case for saturated or unsaturated at this point. Yeah. Yeah.
So CCK, is it the one that neutralizing neutralizing the acid or pepsid, or it's something else entirely different?
CCK is all about fat.
Okay.
So CCK is released in response to fat, and then all of its downstream effects are to help deal with fat. So reduce motility and the release of bile and bile salts. And then we have also this sort of lesser role that the pancreas is playing as well. but we'll just chalk that up to more contributions to fat. Okay, at the same time that all this goes on, so these are responses that aren't CCK mediated necessarily, but they would happen with any meal, that the pancreas is also generating secretions that are going to contribute to chemical digestion and absorption. So those include enzymes. And the enzymes like trypsin, collapase, and amylase, those are all secreted by the pancreas. So those are getting dumped into the lumen of the stomach, and they're going to interact with the chyme and travel with it all along the way, doing what they do, taking large molecules and making small molecules all along the way. Then the other enzymes that can work on it, as I mentioned, are the brush border enzymes that are anchored to those absorptive cells all along the way through the gut. So both are acting to cleave chemical bonds to make small molecules that can be absorbed. All right, so let's consider carbs, proteins, and fats. And that's kind of the level of complexity. It gets sort of increasingly complex in how the chemical digestion occurs and then the absorption that occurs as a consequence of that. So first, carbs. So we have here a glucose polymer, okay, like glycogen or starch. And the first enzyme, and this one, a lot of the enzymes that matter here, I'm not going to lay on you. I'm not going to make you responsible for them. But there's a few that I will mention. And so amylase is just a big deal. Amylase comes up, it's secreted first in the mouth, and it plays a major role in the chemical digestion of carbohydrates. So we got to mention that one, and it's released by the pancreas. And so it works, by the time it's done its work, then we have, instead of a polymer, a series of disaccharides. So two molecules that are connected together. And these would include okay, so then we have a variety of enzymes that are located on the brush border. And so just imagine on the right, on the right here, I don't want to touch my computer's in the way. I don't want to touch it because that connection was not working when I first got here. So I have to look around. Okay, so just imagine that here on the brush border, we've got a bunch of enzymes. And those enzymes, now that we have the amylase that's floating around in the lumen of the gut and producing disaccharides in our chyme, that food slurry, now these can be broken down further. And the brush border enzymes, I'll just mention them, but you don't have to go home and memorize them. They're maltase, sucrase, lactase, and they are going to act on maltose, sucrose, and lactose. So if you had to memorize them, it wouldn't be that hard if you know what the disaccharides are called. And that yields monosaccharides, so single unit sugars, single molecules like glucose, fructose, and galactose, and I'll just show them as different symbols. So all that you need to be responsible for is what appears on the slide. The things that I'm saying are just for your own benefit. You don't have to go home and memorize my words, but just do know the terms that appear on the slide. All right, so by the end of these processes, we've got amylase in the lumen of the gut, producing the disaccharides, the brush border cells, then producing monosaccharides. Now we're at the point where absorption can occur. Okay, so absorption is, of course, getting the molecule from the lumen of the gut across the absorptive cell and into the bloodstream. And we already way early in this course talked about glucose transport. So we did it more in detail back then than we're going to do now, where we had the primary active transport, we've got the sodium potassium ATPase right there. That's creating a sodium gradient. Then we have a co-transporter that is making use of the sodium gradient, allowing the glucose to enter into the cell, where it can then exit via facilitated diffusion. All right, so the bottom line for carbohydrate absorption is that the carbs are broken down into monosaccharides, and then they're absorbed via transporters. Okay, pretty simple and pretty familiar territory because we already talked about the absorption process. Okay, now protein digestion. So again, big molecules become small molecules. So a protein has this kind of chemical composition. You probably know this from prior courses. You've got an amino terminal, a series of amino acids, and then it ends in a carboxy terminal. Okay, so when I was talking about the action of pepsin, it's breaking these peptide bonds between amino acids. And it's an example of what we call an endopeptidase. So an endopeptidase is a class of an enzyme that's going to break up those peptide bonds on the interior of the molecule, highlighted there in red. So that's going to yield small peptides. And if you got 3 amino acids in the middle, you call it a tripeptide. And then if you cleave the bonds that are connected to the amino or carboxy terminal, then your enzyme is called an exopeptidase. So endopeptidases include also trypsin in addition to pepsin, whereas exopeptidases are things like carboxypeptidases. So that's cleaving these outer bonds so that you end up with amino acids and dipeptides. Okay, so all 4 of those, tripeptides, small peptides, amino acids, and dipeptides, can be transported. Can be absorbed. All right, so let's consider the first. So just amino acids. That transport is very similar to what you see for glucose. So again, making use of a sodium gradient and co-transporting via secondary active transport, followed by facilitated diffusion. Okay, tripeptides and dipeptides are transported in a similar way. It's just that instead of using sodium, they're transported with protons. All right, so something similar. There's a gradient of protons, secondary active transport. They make their way into the cell. Okay, small peptides are more of a problem because they're so large that these mechanisms don't seem to work. And so there's this trick that absorptive cells have where they create a vesicle around the whole molecule and essentially produce this internal bubble that gets transported through the cell. So it never actually mixes with the cytoplasm of the cell. And we call that transcytosis. And so it just kind of goes through like in a force field, making its way through the cell. Okay, so protein absorption is sort of like carb absorption, but with a little bit of a twist because you have slightly different handling for the di and the tripeptides, and then the larger proteins have to go through by transcytosis. All right, fats are a different story. So if we imagine that the fat droplets that emerge from the cheeseburger at this point are sort of like the olive oil in a salad dressing if you shake it up, you've got these large globules of fat. Now, a large globule is a problem in terms of the surface to volume ratio, right? You want this whole volume of molecules to be absorbed, but the enzymes can only act on the surface. And so it's much better to have small droplets like you can create if you really vigorously shake that salad dressing. And in order to facilitate this partitioning of the volume into small droplets, that's where the bile salts come in. And what the bile salts do is they essentially coat the layer of the droplets. And so as the stomach churns and effectively mixes that salad dressing, then the bile salts come in and coat the surface of the salts, of the globules. And that makes the small volumes stable, so they don't just reform into large globules. So the bile salts that were released by the gallbladder, that were manufactured by the liver, are around in this slurry in the stomach. And as the large globules are shaken up, then they stabilize these small volumes. And these small volumes contain potentially a variety of fats, including triglycerides and cholesterol. And now is when we have enzymes that act on these molecules to produce even smaller molecules. fatty acids, and monoglycerides. So it's in those forms that we have the absorption of fat. So the way that they do this is, again, because we have this phospholipid bilayer that is very sort of permissive for fats to go across the cell membrane, the monoglycerides and the free fatty acids just diffuse into the cells. Okay? They don't need any special carrier proteins in order to make their way into the interior of the cell. The same is not true for cholesterol. That does require a transporter. And so we have a transporter that does just that. Okay, once they're on the inside of the cell, then they fuse with the Golgi apparatus. And so they're handled on the interior of the cell in a different way from the carbs and the proteins. And then they're also handled differently as they're transported through the basal side of the cell, because they're passed off into those vessels, the lacteals, where they're transported through the lymphatic system. And with the time that remains, we've got one very quicker question. If your body ceased to produce bile salts, what would happen? A, protein would have to be absorbed as small peptides. B, fat globules in the chyme would remain large. C, you would defecate a relatively low concentration of fats. D, you would crave salty foods. Or E is B and C. Okay, give me your final answer. Okay, and the correct answer is B. Okay, see you next time.